Hi everyone, we're going to be moving on to chapter 11, which is all about cell communication. Cells needing to speak with one another. It's a great question, right? How do cells talk to one another? Well, cell-to-cell communication is essential for both multicellular and unicellular or single cellular organisms like prokaryotes. Think about it. Think about communication. Think about how you communicate with other individuals. You can communicate a message that's close by, like to someone standing next to you, maybe passing a post-it note or just speaking and saying something. You can communicate to one person from maybe a long distance, like with an email, or you can communicate to tons of people at once, such as sending out a Facebook post or an Instagram post, right? There's many ways to communicate, and cells have those same obstacles to overcome, whether it's going to be a close by message to some cells that are nearby, or to different cells that are way, way far away from where they're located within the body. And even prokaryotes who don't even have a brain and they're simple cells without a nucleus are able to talk to one another. And yeast can talk to one another, who evolved 1 billion years before humans did. It's amazing how they can literally talk to one another. And biologists have discovered some universal mechanisms with how they can speak to one another, and it allows them a way of regulation. The way cells talk to one another is by using chemicals. Now, those chemicals generally are hormones, which are lipid-based, or they can also speak to one another with proteins as well. And so within our chapter, I want us to keep going back to this. This is an amazing thing to think about. It's the fight or flight response, or what you might also know of as like an adrenaline rush. Another name for adrenaline that scientists use for it in the body is called epinephrine. And imagine what a gazelle must do to outrun a cheetah. Number one, it's going to need a lot of ATP, right? And it's going to have to coordinate many, many cell responses in order to run at, who knows, 60, 70 miles an hour to outrun that cheetah darting back and forth. It's got to get all those cells working together. It's got to get all those cells making ATP. But remember, we don't store ATP. You can only literally make it on the fly. So in order to make that ATP, you need glucose. And you only have some short-term storages of glucose in muscles and the liver. You literally need to get glucose. You can't stop and just have a snack there quick of some sugar. That's not going to fly. So you're going to have to break that glucose, or you're going to have to make that glucose from glycogen stores in the muscles and liver and elsewhere in the body. And that's one of the things that epinephrine does, is that it activates a response in cells, or it tells cells, it communicates to cells to tell them to start breaking down glycogen to make glucose, so you can quickly make ATP, so you can outrun that cheetah. Yeah. Outrun the gazelle, outrun the cheetah. So let's keep going back to that as we think about cellular communication. And as I said before, microbes evolved the ability to communicate with each other even before us, billions of years before us. And when we look at how they communicate with one another, it's very similar to how our own cells communicate with each other. So we'll take a look at that. So one of the first studies that we'll take a look at here is yeast. Saccharomyces, do I dare say that other word, cerevisiae? have two mating types. They have A and alpha mating types. And how about I get a picture here? There they are. Here's the A type, and here's the alpha type. And so the A type needs to get together with the alpha type in order to mate. Sounds pretty reasonable, doesn't it? But how does the A and the alpha know that, number one, they're there, they don't have eyes to see each other, or a brain, or a mouth to talk to one another, so they use chemicals. So we say that A yeast releases A factor, and the alpha yeast releases alpha factor. Now it turns out that the opposite yeast cell has the correct receptor to receive that signal from the other type. So the alpha factor from the alpha yeast fits into the receptor of the A yeast, and then vice versa. The A factor from the A yeast fits into the receptor of the alpha yeast. That way, number one, they know that each other is there, and they know that they're able and ready to mate with each other. So they release those factors, then they're received, they merge like you see in the picture. That's their mating, basically, and that merges their genetics together so they can get together and have all the genes and benefits that their descendants will be needing from here on out. Isn't that kind of romantic, right? So this involved what's called the signal transduction pathway. This is a series of steps by which a signal on a cell surface is converted into a specific cellular response. Sometimes these signal transduction pathways involve just one step, and sometimes they involve many steps with many relay molecules in between. It all depends what the organism is and what the cells are and what the desired response will be. So signal transduction pathways then convert those signals on a cell surface into cellular responses. These romantic yeast here. And these pathways that they can do this and the chemicals that they use have similarities and suggest that an ancestral signaling molecules evolved in prokaryotes and then later were modified in eukaryotes. And the concentration of signaling molecules allows bacteria also to talk to one another and to sense how many of them there are around them. Like, for instance, here's some bacteria that live in soil. So they can release a chemical, and when there's only a few of them, there's not enough chemical. But when there's enough of them, then there's enough chemical that they know how many are around there, and they can initiate their response then. Such as, let's think of it when a bacteria invades our body. You're a big organism, and one bacteria releasing some chemicals really isn't going to have an effect on you. So bacteria are able to wait and reproduce into millions, and each of them are releasing their certain amount of chemicals, and it's talking to one another using those chemicals so that they know when the chemicals reach a certain level that there's enough of them to launch their desired response or attack on you or the organism that they've invaded. That's called quorum sensing. We're going to be watching a video on that with a wonderful Dr. Bonnie Bassler, a great scientist who's doing some awesome research on quorum sensing and bacteria. But they're able to talk to each other and know exactly when there's enough of them to launch that attack. Because alone they wouldn't be able to do it, but together they could be strong enough to make an impact and, well, do what they got to do. In this case, in this picture here, these soil bacteria are talking to one another, and they're realizing that there's not enough nutrients to sustain them all, so they're coming together in what's called the spore forming structure and making these fruiting bodies that will take them through these bad times until the conditions become more desirable. So, cells in a multicellular organism communicate by chemical messages, and so those would be proteins or lipid-based hormones. Animals and plants can communicate, their cells can communicate with each other if they're really close by. They have little spaces in between. Let's look at a picture. So first of all, some cells that are very close to one another have what are called gap junctions, and these are little holes in between the cells. The lipid bilayers are connected, and the holes within the cells of neighbors are able to transmit these chemical signals just from one cell to another relatively quickly. Plant cells have something similar. It's tubes that connect the cells, that go through the cell membranes and the cell walls and connect the cells. Those are called plasmodesmata, and they can transmit signals through those as well. Or how about this? Two cells that are very close to one another. One has a certain protein on the outer surface, the other has another, and they fit together like a lock and a key to recognize one another and transmit a signal. So this is called local signaling. It's the ability of animal cells and plant cells to communicate by direct contact or cell-to-cell recognition. In many cases, the animal cells use what are called local regulators. Those are messenger molecules that just travel short distances. But in a lot of other cases, some of the signals have to get further, so hormones are used. The ability of a cell to respond to a signal actually depends on whether or not the cell has a receptor that's specific to that signal. If it does, then we would say that that cell is a target cell. Such as, let's look at some examples here. This one right here, this is more local signaling. This is called paracrine signaling. And it's very similar to like the endocrine system in using chemicals to signal cells, but in paracrine signaling, the signal is local. It's signaling cells that are very close to you. But in endocrine signaling, the signal travels usually a larger distance. In paracrine signaling, this cell is secreting its chemical message to the target cells nearby. They have receptors to receive that signal, so they will respond to the signal. Any cells nearby that do not have the receptor, they'll ignore that message. Here's another example of local signaling. Remember, neurons are brain cells, and no neurons touch each other. You wouldn't want to get your electrical wiring crossed, right? So there's always a space between neurons. That space is called a synapse. So this is called synaptic signaling. We don't want the message to stop. We want it to keep going. And since neurons don't touch each other, the signal has to go from electrical to chemical by traveling across that gap or that synapse. So chemicals called neurotransmitters are used. As the electrical signal comes to the end of this neuron, it causes these vesicles to release their neurotransmitter into the synapse, where then they're received by receptors on the next neuron, which then transmits the signal back to electrical and keeps it going. The neurotransmitters then are, any that are left over in between the synapse are quickly broken down. All right, going a longer distance here. The endocrine system is a great example of having to have cells communicate, like maybe in the brain, with cells that are way, way away from that. So the blood vessels, the circulatory system, is used as a method to transfer that signal. So in this case, we have an endocrine cell, and it's made its hormone, it's released it into the bloodstream nearby, and it travels through the bloodstream to what's called a target cell somewhere else. The target cell has receptors for that signal so it will respond to that signal. Otherwise, all the other cells ignore it. That's long distance signaling. All right, so in review, we took a look at some examples of local signaling, like going through gap junctions and plasmodesmata and cell-to-cell recognition, paracrine signaling, and synaptic signaling. And then also we looked at long-distance signaling, where signals travel long distances. So there's been some research on this. Sutherland is one of the scientists heavily researching these areas right now, especially the action of epinephrine on cells. So Sutherland suggested that cells receiving signals went through 3 processes. I want you to commit these 3 to memory in the order that they happen. First of all, reception happens, then transduction, and then response. Let's take a look at what that might entail. So here we have a cell. I'm in the cell. I'm going to make myself the nucleus here. And this is the cytoplasm, the plasma membrane or lipid bilayer, and then we have a protein receptor in that lipid bilayer. Here comes the signaling molecule. A signaling molecule might be a hormone, that might be lipid-based, or it could be a protein. Either way, the signaling molecule is called the ligand. It bonds into the receptor like a lock and a key. We'll say this cell is indeed a target cell for this message. So this is called reception. The target cell detects the signaling molecule that's coming from outside of the cell. So reception. All right, number 2, transduction. Now that our receptor protein has received the signal, often that causes the receptor protein to change, often changing shape, which then initiates transduction. This converts the signal to a form that brings about a cellular response. Whatever happens here in transduction, it might be just one step, and it could be a number of steps, like you see this signal, these chemicals changing form from one thing to the next until we get to our final thing. Again, these are called relay molecules that are in the sequence of changes here in transduction. But until we get to number 3, our response, which is an activation of a cellular response. This could be activating an enzyme that's in the cytoplasm. It could be something as simple as that. It could also mean activating what's called a transcription factor that goes into the nucleus of the cell, reads a gene, causes the reading of a gene, and causes that gene to be transcribed into a protein. So it could be something more complex like that. In our example with the cheetah and the gazelle, we're going to use the model of what epinephrine does. How epinephrine is the signaling molecule, the ligand that bonds to the receptor, brings about these series of changes, and then gets to our ultimate response. What's our ultimate response with an adrenaline rush or epinephrine? Well, it's the breaking down of glycogen so that you can have glucose going through the blood and going to cells and being used to make all that ATP that's needed to outrun a 60-mile-an-hour cheetah. Going to need a lot of ATP for that. So if you take a look through your book, you're going to see there's a bunch of different types of reception, number of ways where cells are target cells and receive a signal. This usually involves, again, the binding of a molecule, what's called the ligand. That's the signaling molecule or the messenger. Then that usually involves a shape change in the receptor protein that's on the cell membrane. And that's often then your lead into the transduction part. And then most signal receptors are on the plasma membrane, but some of the receptors are actually in the cytoplasm. If the actual signaling molecule is a lipid hormone, that's actually able to go right through the plasma membrane, because the lipid hormone is made out of similar molecules as the plasma membrane, lipids, phospholipids, and so they're able to dissolve or diffuse across that membrane and find its receptor in the cytoplasm. But as it says here, most signal receptors are plasma membrane proteins, and the ones that receive epinephrine or adrenaline are actually plasma membrane proteins, which is hard to say. All right, so most water-soluble signal molecules bind to specific sites on receptor proteins that span the plasma membrane. And there's three main types of membrane receptors: G protein coupled receptors, or sometimes just called G proteins for short, receptor tyrosine kinases, and ion channel receptors. So epinephrine uses the G protein receptor. So I want you to take a look in your book and read about how they work. They have some pages with some wonderful diagrams dedicated exactly to these types of receptors. I want you to look at the pictures and follow the sequence of events that happens in all of these. The G proteins, the receptor tyrosine kinases, take a look at those. You'll see some wonderful diagrams on how those work. See if you can picture it happening as it goes. And then the ligand gated ion channels. Take a look at those as well. Those might be a little easier to understand in things going through a protein channel. It's maybe similar to when we did the models of the sodium-potassium pumps. So take a look at those. We'll be looking at them a little more specifically, but we'll go into the detail here today. All right, then we'll also take a look at some examples of when in cytoplasm receptor, like in this picture here, is used. So we'll use that. And then we'll go into some of the transduction examples. It usually involves multiple steps. One of the steps might amplify a signal. So instead of just making one thing, you make many of them, and then they go on to signal many more, and you get what's called amplification. So then you get a lot of product instead of just a little. That's extremely important. And let's see here. And in many of the pathways, the signal is transmitted to get phosphorylation to happen, right? Phosphorylation is the adding of a phosphate group to a molecule to destabilize it and activate it and get it primed to do work. So protein kinases are the enzymes that transfer phosphates from ATP to a protein, and that's called phosphorylation, as you already know. Protein phosphatases are the opposite. They're the enzymes that remove the phosphate groups. So many of these transduction pathways involve protein kinases, where they phosphorylate a molecule, or you get amplification and you get many molecules phosphorylated, and protein phosphatases take away those phosphate groups. Take a look at that example there. Look for that one in the book. See if you can follow the sequence of events that are happening. And then we should also know the names of a few other molecules involved. One of these is called a second messenger. And the actual first messenger would be your ligand or your signaling molecule that initially started all of this. The second messenger is another molecule that's usually in the cytoplasm of the cell, and then is used to activate other molecules. So a very popular second messenger is a relative of ATP. Remember, ATP stands for adenosine triphosphate. Has 3 phosphate groups. Well, there's a relative called adenosine monophosphate, meaning it only has 1 phosphate group. So adenosine monophosphate, let's see if we can get a picture here. There's ATP with its 3 phosphate groups, but here's AMP, adenosine monophosphate. It just has the 1. And sometimes this phosphorus group will kind of piggyback and bond up here to this sugar. So then it creates this look. So then we call it C-AMP or cyclic AMP or cyclic adenosine monophosphate or CAMP. Goes by quite a few names and looks like this. But basically just think of it as ATP missing a couple phosphate groups. All right, don't make it too complex there. But that's what it looks like, and it's often used as a second messenger, one of the most widely used ones. Another molecule we might need to know the name of is, I hope I can say this right, adenylyl cyclase. Adenylyl, there we go, adenylyl cyclase. This is the enzyme in the plasma membrane that converts ATP to cyclic AMP in response to a signal. So this would be part of that transition pathway, making cyclic AMP molecules, because cyclic AMP molecules are important in that they're the ones who activate the protein kinases, which then go on to phosphorylate enzymes, which then that enzyme does work and makes our product for us, right? So look at that that pathway there. We go from adenylyl cyclase, which is often, there it is, in the membrane. After adenylyl cyclase gets activated, then it takes a ATP and makes it into cyclic AMP, and then cyclic AMP goes and activates a kinase, and then kinase brings about whatever cellular response that you want. Whew. That's a pathway, that's a transduction pathway for you. So a lot of times it's not just one step, it's quite a few steps. Calcium ions are often used as second messengers as well, rushing in of calcium or absence of calcium. So that's another example to look out for as well. And again, I mentioned some of the different, for part 3 of this, different responses might be activating an enzyme to get a product or activating a transcription factor to go in and find a gene in DNA and then cause RNA to transcribe it and then making a protein from it. So that could be another response. One final response that I want to show you. It's called, I just love this word, but it literally means cell death. It's apoptosis. Apoptosis. It's programmed cell death. It's cells that have a receptor to receive a signal to literally commit suicide. This is when cell components are chopped up, packaged into vesicles so they can be digested by the scavenger cells like macrophages. This prevents enzymes from leaking out of the cell and then destroying nearby cells. Sometimes a cell goes bad, let's just admit it. Sometimes a cell makes bad proteins or has damaged DNA or is just run down. So it needs to go. And so the receptor here receives the signal, which then here's our signal transduction pathway. We don't need to memorize all these steps, but the end response is making these enzymes, proteases and nucleases, which basically cut and chop things up to get it packaged to be able to get rid of it, right? That might look something like this. All right, and sometimes it's important even in like development in fetuses and embryos. you actually had to have cell death happen in between your fingers to get rid of that tissue so that you could have fingers that weren't webbed. All right, so in review, we just did some general things here with reception, transduction, and response, and we'll be looking more specifically at some models of this. And there'll be a lot of different things that happen in this transduction. We're going to look at the big picture usually. And don't get too overwhelmed by the details and the names of all the different little molecules that are involved. Again, always think, what's the big picture going on here?